Hello everyone, welcome back to PhytoPathoVerse. I'm Malini, molecular plant pathologist with a motto to make plant pathology easy. So today we are going to start with chapter 4 of JN Agrios, which is genetics of plant disease and resistance. So in chapter 4 we will get to see the involvement of genes in plant diseases, how genes play a role in managing plant disease, in making it resistant, or whether to make it susceptible. Okay, because genetics, what is it? It is the study of genes. Therefore, in this chapter we will come across many terms that we have studied maybe in our plus 2 or in biology, and who loves genetics, it will be an easy thing for them. But for the ones who don't, and they don't have that idea about what is gene, DNA, RNA, introns, extrons, ATGC, nucleic acids, so we will try to cover it one by one and line by line. Okay, let's start. The genetic information of all organisms, that is the information that determines what an organism can be and can do, is encoded in its DNA. Yes, for all the animals we have DNA. Yes, in RNA viruses, of course, it is encoded in their ribonucleic acid, which is RNA. Yes. So in all organisms, most DNA is present in the chromosomes, and in prokaryotes such as bacteria and molecules which lack an organized membrane-bound nucleus. What does that term called? The nucleus which is not having an well-organized bound membrane, it is called nucleoid, not nucleus, which means primitive nucleus. Okay, which we get to see in case of bacteria, prokaryotes. Okay, so there is only one chromosome and it is present in the cytoplasm, whereas in eukaryotes there are several chromosomes and they are present in the nucleus. Alright, many prokaryotes, however, and some eukaryotes also carry smaller circular molecules of DNA called plasmids. What is plasmid? Plasmid is the extra chromosomal DNA. Again, where we get to see this? In case of bacteria, okay, in the cytoplasm. And plasmid DNA also carries genetic information but multiplies and moves independently of the chromosomal DNA. Plasmid is used as a vector for transmitting genetic information from one organism to another, right? And that method is also called horizontal gene transfer, that we are going to study in the coming sections, right? So plasmid DNA also carries genetic information, already covered. Furthermore, eukaryotic organisms carry DNA in their mitochondria. Plant cells, in addition to nuclear and mitochondrial DNA, also carries DNA in their chloroplast. So sometimes we get such questions that normally we see DNA in nucleus, but other than nucleus, what are the other cell organelles in which we see or which possess DNA, right? So for eukaryotes we see DNA in mitochondria, and in plant cells it is also have in chloroplast, right? So genetic information in DNA is encoded in a linear fashion in the order of 4 bases, which is A, C, G and T. A for adenine, C cytosine, G guanine and T thymine. Now can you tell me what DNA is made up of? The components of DNA are nucleotides, that are the basic building blocks of DNA, right? The phosphate group is what? A molecule that links the nucleotides together. Next, the what? Deoxyribose sugar that we have seen here. Deoxyribose sugar, okay? It is a 5-carbon sugar that forms the part of the DNA backbone, and the one that we have seen just now, the bases, nitrogenous bases. It's of 4 types: adenine, cytosine, guanine and thymine. Now there are more functions of DNA, and there are structure of DNA like sugar, phosphate, backbone, base pairing. Base pairing is what? Like A is paired with T, adenine with thymine, AT, G, C. Guanine is linked with cytosine. A and T is linked with double bond, and C and G is linked with triple bond. Okay, and they have their specific functions, right? So the DNA is a totally different lecture if we start with. So moving forward, each triplet of adjacent bases codes for a particular amino acid. Each triplet means what? So triplet means 3. It means ACG, TAC, AGT, like this triplet. So each triplet, if we make it adjacent, we bring it together, so it codes for a particular amino acid. Amino acid, it is the part of protein. Okay, the code is redundant such that 61 triplet codons code for 20 amino acids, and 3 triplets are stop codon. 3 triplets are stop codon that terminate protein biosynthesis during messenger RNA translation. Now there are start codon and stop codon. Let me write it for you. Okay. So there are start codon from which the synthesis starts. Okay. So start codons are A, U, G. Whereas this is the start codon, right? Whereas the stop codons are, there are 3 stop codons, which is UGA, UAG and UAA. Okay, so these are the 3 stop codons that signal the termination of protein synthesis or translation, right? Again, we have heard of the term called central dogma, in which there are 2 steps. One is transcription and translation, right? Transcription is the conversion from DNA to RNA, mRNA, right? Or translation is what? The conversion of RNA to proteins, right? So this stop codon, it signals and stops the translational process by what? By binding to release the factors on the ribosomes. Moving forward, a gene is now what is gene? We will see what is gene. It is a stretch of a DNA molecule that encodes a specific function. So gene is nothing but a stretch of DNA itself, okay, which encodes a specific function. It usually codes for 1 protein molecule or less, frequently 1 RNA molecule as a functional unit. In eukaryotes, what happens? The coding region of a gene is often interrupted by a non-coding stretches, and that is called introns. See, if we look into this diagram, what they are saying, the location and arrangement of genetic material, A, eukaryote. So in eukaryote, we will see DNA in nucleus, mitochondria and chloroplast. Obviously chloroplast is present in plant cells. And what will happen in prokaryotic cells such as bacteria? There will be DNA we can see only in chromosome and plasmid. Okay, and for plasmid it will be extra chromosomal DNA, and it is circular in shape, right? Okay. Now, in many cases of plant pathogen interaction, gene is one organism are triggered to be expressed by a substance produced by other organism. Let's see one example. So genes for defense reaction in the host, example the production of antimicrobial secondary metabolites called phytoalexins. Now, if anybody know was any examples of phytoalexins, or what is the definition of phytoalexin, okay, please let me know. And apparently are triggered to expression by certain signal compounds activated by inducer molecules, which is elicitor produced by the pathogen. These terms, elicitor, effectors, all these are very important terms which normally get confused, but it's very important when we will study the host pathogen interaction or the defense mechanism that that happens in plant cells by attack of any pathogen, right? Also the production of phytoalexin. Yes, I have already told the production of phytoalexins. So there are basically two terms, one is phytoanticipants and another is phytoalexins. So when there is pre-existing defense mechanism, that is phytoanticipants, whereas phytoalexins are the post one, after the defense that is created after the pathogen attacks the host. There are examples. Go search. If you can't, then please let me know. All right. So such a sequence of events requires the function of several host genes. The genes encoding for a receptor that recognizes the elicitor and translates the pathogen signal into a host signal. So in computer we have heard, right, the human language, the machine language. Machine language is the 0, 1. Yes, we have heard it in schools. So here what happens is that the pathogen, when it attacks, they release some signals, but that signals needs to be recognized by the plants, right? If it gets recognized, then they will start, or they will prepare their, or they will build their defense against those pathogen. If it cannot recognize the signal which is transmitted, which is coming from the pathogen, then what happens? Then the disease will cause, right? So there is no recognition, means disease will occur. Okay, so there are few genes which encodes for a receptor that recognizes these elicitors and translates the pathogen signal into the host signal. So the genes for signal transduction protein that transfer the signal into host nucleus. Genes for inducible transcription factors that are activated in the presence of the elicitor, and the genes expressed under the control of such inducible transcription factors that codes for the enzymes that cooperatively synthesize the phytoalexin. All are the works of separate genes. Genes and genes. All are the works of genes, okay? But separate one, remember? Genetic determination of plant disease. In nature and in crop production systems, different plants show different occurrences and degrees of plant disease when confronted with a specific pathogen. Indeed, plant diseases only occur when a specific pathogen that is adapted to a specific host plant contacts the plant. The same pathogen may cause little or no disease when confronted with a different plant species. So what do we understand with this this till now is that, for example, Puccinia graminis Pharma specialis triticide, right? Triticide is wheat, and the pathogen causes what? Stem rust of wheat. So this is very specific to wheat, but not other crop like brinjal or rice. Okay, so they are specific. And if this pathogen attacks rice crop instead of wheat, so there will be no effect on rice crop. That's what they are telling. Okay, so being a pathogen does not generally mean being effective in infecting many diverse plants. So yes, pathogen, so they are the bad organism here who attacks the crop and make them diseased. But it's not like pathogen A, if it attacks wheat, like Puccinia graminis triticide, if it attacks wheat, then it is harmful to wheat and wheat only. If it attacks rice crop or some other crops, solanaceous crop or Fabaceae family crops, so there will be no effect on that. Okay, even if a pathogen is adapted to a specific host plant, not all plants show disease. And if they show disease, not all plants show the same severity of disease, right? So they have different severity level to cause. Means now here we will understand the difference between pathogenicity and virulence. You know, we have heard of these two terms. So pathogenicity is the ability, the inherent ability of an organism to cause disease, right? Till that clear. But when we talk about virulence, now virulence is the degree. Okay, so see, this is what we are talking about. Severity level, the degree of severity. Okay, so virulence is the degree of severity or ability of a pathogen to cause disease. Now, if a pathogen has a narrow host range, we often observe that some host genotypes show complete resistance to some pathogen isolates or races to this species, because while the same plant genotype is often susceptible to other races of that same pathogen species. This kind of resistance is called race-specific resistance. And race-specific resistance is also called as what? We have heard of vertical and horizontal resistance, right? So race-specific resistance is the vertical resistance, which is very specific, and with this we can gain complete resistance over that pathogen, because it is governed by one or very few genes. So it's possible for us to to gain that complete resistance. But when it is horizontal resistance, that means it is not host specific and it is governed by multiple genes. So taking over that multiple genes is very difficult. But in case of race specific, fewer genes, so it's possible to gain complete resistance. So vertical resistance is the race specific resistance, complete resistance, but they are unstable and can be governed or overcome by new pathogen strains, right? Whereas in contrast, horizontal resistance is partial, because it is governed by multiple genes and they are non-specific but stable resistance, yes, and it provides broad spectrum protection against all pathogen races. Now we will move forward to variability in organisms. One of the most dynamic and significant aspects of biology is that individuals within a species are not uniform but vary from one individual to another, right? We all are different. We all are same but different. There will be variability, right? in a single class. All are students in the same class and around same age, but they have variations. The students, there are various types of students who are more intellectual, who are more hardworking. Okay, even in the teachers, there varies, means everybody is different, same but different. As a matter of fact, all individuals produced as a result of sexual process, such as the progeny of one animal family, are expected to be different from one another and their parents in a number of characteristics, although they remain most similarities with them and belong to the same species. Yes, this is also true for oomycetes and fungi produced from sexual spores such as oospores, ascospores and basidiospores, of parasitic higher plants produced from seeds, and the nematodes produced from fertilized eggs, as well as cultivated plants produced from seeds. So even bacteria have mechanism for the transfer of genetic information. When individuals are produced asexually, the frequency and degree of variability among the progeny are reduced greatly. But even the individuals among the progeny will often show significantly different characteristics because of the astronomical number of individuals. What is this astronomical number? Means large, like greatly large individuals produced by microorganisms asexually, and the short generation times during which they are subjected to selection in different environment, the total amount of variability produced by at least some organism is probably as great and possibly greater than the total variability found in microorganisms produced only sexually. I hope this is totally clear, right? In sexual reproduction, the process is very slow, whereas in case of asexual reproduction, like budding, fission and fragmentation, all those asexual reproduction, there are numerous progeny that we see in very short duration. Okay. So the genetic variability, however, in a gene pool of a pathogen population determines its potential to quickly adapt to new environments such as changing weather conditions during global warming, or new plant varieties such as disease-resistant cultivars. The understanding of host and pathogen genetics and the diversity of compatible and incompatible interactions is therefore fundamental of plant protection that relies on host genes and biotechnology. Mechanisms of variability. In host plants and pathogens such as most fungi, parasitic higher plants and nematodes, which can and usually do reproduce by means of sexual process, variation in the progeny is introduced primarily through segregation and recombination of genes during the meiotic division for the production of sexual reproduction cells that derive from zygote. I hope we are familiar with the sexual reproduction. In genetics, the Mendelian steps, that is segregation, recombination, we have studied. This explains why the progeny of sexual reproduction is usually more variable than the progeny of clonal propagation during asexual reproduction. See, with the name itself, clonal propagation. Clone means what? Clone is the ditto copy. Means if I make a clone of myself, it will be the ditto copy of me. So how I talk, how I react, all my emotions, all the genes, the character, everything will be cloned. Means photocopy, right? Whereas in case of sexual reproduction, there is this segregation, recombination of genes, addition, deletion, mutation, so many things that happens in sexual reproduction. So there are high chance of variability. In bacteria too, and even viruses exhibit variation that seems to be the result of recombination. In many fungi, heteroploidy and certain parasexual processes lead to variation. Okay. However, all plants and all pathogens, especially bacteria, viruses, fungi, and molecules, can and do produce variants by means of mutations in the absence of sexual spores. Now we will check few of the general mechanisms of genetic variability, starting with mutation. So we normally see two mechanisms of variability, one mutation and the second is recombination, occurring in both plants and pathogens. In mutation, what is it? It is a change in the genetic material of an organism, which is then transmitted into in a hereditary fashion to the progeny if it is not lethal. In addition, somatic mutations occur in non-reproductive cells of multicellular organism, often remain unnoticed but can also lead to somatic alterations such as pigmentation changes. We have heard of reproductive cells and non-reproductive cells, right? Reproductive cells are the ones which helps in reproduction, sexual reproduction. In case of humans, what do we know? The sperm cells, right? They are the reproductive cells which helps in the sexual reproduction to form zygote, right? But what are non-reproductive cells? They are the somatic cells. Okay. So even in somatic cells, there can be alterations, okay, such as pigmentation changes. Now, mutation represents changes in the sequence of bases. What are bases? Before some time we have discussed bases means A, T, G, C, adenine, guanine, cytosine, thymine, right? So, there will be some alterations in the bases in the DNA, either through substitution of one base or another, or through addition or deletion of one or many base pairs, right? If we have like it's normal, but it's like A, T, G, C, C, G, T, A, something like this, and something got deleted. Instead of this, it's substituted with some L, some another base, or we can add one more base. Okay, there, so there will be either addition or deletion or substitution. These are the mutation, point mutations, right? There are so many types of mutation, you know, point mutation, frame set mutation. So these are the types. Okay, moving forward, additional changes may be brought about by the amplification of particular segments of DNA to multiple copies by insertion or excision of a transposable element, that is a movable DNA fragment, into a coding or regulatory sequence or a gene. Okay, so we have understood the main concept, right? How do mutation occurs? This is the simplest way that we can understand mutation. Is these are the bases of a DNA, right? If any of the bases got exchanged or added or deleted, we have a totally new segment of DNA which will code for if this segment of DNA was coding for a black hair color. So if we add or delete something in between, so the character or the trait will get changed, and maybe we get a golden yellow hair color. That is it. Okay. Let's see for the plants what they are saying about mutation. Okay. As plants and pathogens contain genetic material DNA outside the cell nucleus in the form of organelle or plasmid DNA, mutation in the extranuclear DNA are just as common as those in the nuclear DNA. Okay, and affect whatever characteristics are controlled by the extranuclear DNA. The inheritance of characteristics controlled by extranuclear DNA, that is cytoplasmic inheritance, does not follow the Mendelian law of genetics. Okay. So mutation on that DNA are more difficult to detect and characterize. Through mutations in extrachromosomal DNA, many pathogens acquire or lose the ability to carry out a physiological process that they could not or could do before. So yes, before I told you a gene is specific to for a particular trait or character. So any changes, it can change. Like in plant perspective, if they can code for a single pigment to produce green color, for example, and we have this mutation, and because of this mutation they lose the ability to produce that pigment, and so we will not get that color, right? That is it. Or in case of flower, we can say in case of flower, because there are we have seen we do all this grafting and all, right? And because of which so many genetical changes that occurs inside of plants, and instead of white color we get to see light pink, or when we do crossing of artificial crossing or natural crossing, means what? There are genetic changes inside. So instead of a solid pink flower and a white flower, we get a mixture of pink and white. That is it. Okay. So three main types of adaptation brought about by changes in the genetic material of the cytoplasm have been shown in pathogens. Pathogens may acquire the ability to tolerate previously toxic substances, that is fungicide or plant defense compounds, to utilize new substance for growth and to change their virulence towards host plants. Several characteristics of plant are also inherited through extranuclear DNA, including resistance or susceptibility of infection by certain pathogens. Now, I just realized that I was giving examples of humans and maybe horticultural stuff, but I should better give example of pathogens, right? So, we have heard of resistance, disease escape, all these terms, right? When and also there are new varieties that are developing. Why all these new varieties are developing? When we are like we have developed a variety of wheat, for example, okay, for stem rust we have developed a resistant variety. Previously this Puccinia could attack the wheat plant. Then with all these plant breeding and approaches, we made a variety of wheat with some genetic changes. After that, this pathogen cannot attack or cannot cause disease to that wheat variety, and they are not compatible, so they cannot cause disease. Now what happens after a point of time, after a few years or some, this pathogen break that resistance in that wheat and they gain the ability to attack that variety. So we have to work on it and again develop one more resistant variety. Coming to recombination. Recombination occurs primarily during the sexual reproduction of plants, fungi, oomycetes and nematodes whenever two haploid nuclei which have been formed after meiosis and contain genetic material that may differ in many loci unite to form diploid nucleus in a cell called a zygote. So the zygote gives rise to a diploid organism, or sooner or later divides during meiosis and produces new haploid cells. Recombination of genetic factors occurs during the meiotic division of the zygote as a result of mainly two processes. Homozygous parental chromosomes consisting of two identical, they had just discussed about the process of recombination, okay, nothing more. Recombination, with the name itself you can say combination of two haploid
what? Nuclei. Haploid nuclei means what? Normally we get to see 2n, but in case of haploid, there is single n, 1n, that is haploid. And when they will n plus n, it will form diploid, 2n nucleus. Okay. Okay, moving forward. What are the other types of genetic variability? Other types of. One, mutation. Second, recombination. What are the other one? Let's see. Besides classical mutation and sexual recombination, other types of genetic variability occur in nature. Yes, for instance, genes can be exchanged by horizontal gene transfer, HGT. Anywhere you will see this written HGT. This is horizontal gene transfer between species, a process for which the mechanistic sequence is often not well understood, but which results in a transfer of one or several genes from one species to another. Such events become evident in pathogens and plants when the transferred gene is of high selective advantage of the receiver organism. Okay. Okay, so I'll just tell you horizontal gene transfer in a easy way. Okay, horizontal means what? When can we do this horizontal gene transfer? Is it possible that horizontal gene transfer is done from mother to its progeny? No, it is only possible when it is only possible in single season. Okay, when two of the crops are grown or cultivated in a single time frame, only then it's possible. Horizontal, that's why the name is horizontal. Vertical means transfer of genes from its mother to its progeny. So what is this horizontal genes transfer? It is a process of an organism acquiring genetic material from another organism that is not its direct parent or offspring. When not human example, plant, when a plant parent, the gene is transferred from parent to its offspring, then it is vertical gene transfer. Why horizontal? When this parent and offspring of another parent, they are not directly linked. In that case, we can do horizontal gene transfer. Okay, and it facilitates adaptation and evolution. It is common in bacteria. Yes, for bacteria, in time of variability, we have talked about this, right? The mechanism of horizontal gene transfer we normally get to see in conjugation, transformation, transduction. These are the variability in case of bacteria. Conjugation, I'll just tell in a short way. Conjugation, it involves direct physical contact between two cells by a sex pili or pilus. Okay. And in case of transformation, what we see? Involvement of environment. So it is a process where recipient cell takes a fragment of DNA from the environment, often from dead or degraded bacteria, and it gets transformed. In case of transduction, what happens? The transfer of genetic material from one bacterium to another via a bacteriophage. Now, bacteriophage is what? A virus that infects bacteria. There are lots of terms, yes, but we will come across all slowly. So that is horizontal gene transfer. Okay. What is this epigenic factor? Let's read. Epigenic factor can also influence plant and pathogen physiologically and hence disease and resistance. How? It is in a simplified way, we can see epigenetics as a process that does not change the DNA sequence of a gene or chromosomal area, but its accessibility for the transcriptional machinery that allows for gene expression. So in this type, they do not change any gene, no addition, no deletion. But what they do? They do something that its expression, means gene has to express itself, right? Means the result. So they somehow change the outcome. So what happens here? The environmental factors provoke physiological adaptation by effects including epigenetic, genetic changes. Gene and genotype flow among plant pathogens. In plant pathology, first, what is gene flow? It is the process by which certain alleles, what are alleles? Alleles are the contrasting character of a particular gene. So in a single gene, again in genetics, we have seen this capital T, small t, right? We have seen this while do crossing. Okay. Capital T, small t. So this is one gene. But in single gene, we have two contrasting character. If capital T encodes for the tallness, then the small t will be for dwarfness. So they are the contrasting character, the opposite character for the height. So gene encodes for the trait which is height, but height of a plant can be higher or it can be shorter, right? So for the tall plants, this is capital T, this is single allele, and for the dwarfness, this is small t. For the this is also allele. So alleles are the contrasting character of a particular gene. This is one of such example. Okay. The process by which certain alleles move from one population to another geographically separated population. In plant pathology, gene flow is very important. Why? Because it deals with the movement of virulent mutant alleles among different field populations. High gene flow in a pathogen increases the size of the population and the geographical area in which its genetic material occurs. Therefore, pathogens that show a high level of gene flow generally have greater genetic diversity than pathogens that show a low level of gene flow. Okay. In pathogens reproducing only asexually, in which no or little recombination occur. Why? Because again we have discussed in asexual reproduction, that is a clonal reproduction, clone, ditto copy. So there is very little or no recombination at all. The entire genotypes can be transformed from one population to another. This is known as genotype flow. Pathogens that produce hardly and airborne spores or other propagules such as rust and powdery mildew fungi that can spread over long distances and distribute their genomes over large areas, sometimes encompassing entire continents. Entire continents, where did we see? What type of spore? What one example of spore that you can give me which can travel continents? One such example of that is uridiospore that we get to see in case of rust pathogens. Okay, it can travel country, overseas, continents, and very long distance. Okay. So, however, soil-borne fungi, oomycetes, and nematodes move slowly and are present in small areas, and their level of genetic flow is limited. Okay. With all types of pathogens, however, their gene flow can be affected significantly by human agricultural practices. Yes, and by intercontinental travel and commerce. In general, pathogens with a high level of gene flow or genotype flow are much more effective and pose a greater threat to agriculture. Yes, that's it. You have understood what is gene flow, right? Gene and genotypic flow in plant pathogens. They have only discussed it further with some more explanations and examples. Coming to population genetics, genetic drift and selection. The size of a population affects the frequency of survival of mutants and thereby the diversity of genes in the population. Populations of most organisms in a geographic area may not be large enough to ensure that each variant will have progeny in the next generation. So random effects would occur during the transmission of genetic traits to new generation. This is known as random genetic drift. It occurs randomly. It is obvious that it is more likely that the uh, one mutant of the smaller population will be lost than the 100 mutants of the large population. That is, in small population, genetic drift result in loss of rare alleles. So, I'm going to tell you this in simple language, this genetic drift and selection, the natural selection. So, what is it? So, natural selection, it is selected naturally, means an adaptive process where individuals with traits better suited, survival of the fittest, we have heard of it. So the traits better suited to the environment are more likely to survive and reproduce, leading to an increase in favorable alleles. But this genetic drift is something else. So this is non-adaptive and random process that cause changes in allele due to change regardless of whether the trait is beneficial or detrimental. That's it. Simple difference between genetic drift and selection. Coming to the stages of variation in pathogen. This we have to read word by word. So the entire population of a particular organism on the earth, example fungal pathogen, has certain morphological and other phenotypic characteristics in common and make up the species of pathogens such as Puccinia graminis that cause stem rust of cereals. Some individual of the species, however, attack only wheat. Remember, only wheat, only barley, and only oats? Yes, so those examples are given here, and these individuals make up groups that are called varieties or forma or special forms or forma specialis. Okay, this is it. So what are those? Puccinia graminis forma specialis triticii for wheat. Puccinia graminis triticii. Fine. Puccinia graminis hordi, which is for barley, and Puccinia graminis aveni, which is for oats. Examples we have already seen. Even within each special form, however, some individuals attack some of the varieties of the host plant, but not others. Okay. Some attacks other set of host plant varieties and so on, with each group of such individuals making up race. Thus, there are more than 200 races of Puccinia graminis forma specialis triticii like res 1, res 15, res 59 and so on. Occasionally, one of the offspring of a race can suddenly attack a new variety or can cause severe symptoms in a variety that it could barely infect before. This individual is called a variant. The identical individuals produced asexually by the variant make up a biotype. Each race consists of one or several biotypes. The appearance of a new pathogen biotypes may be very dramatic when the change involves the host range of the pathogen. About all this, everything will get clear if you just study this table. You can take a screenshot of the table, or I'll attach it in the description for you. Okay. So for fungi, for bacteria, viruses, nematode plants, they have done it very nicely, like how to distinguish. The first is genus, the level. They have categorized is level wise. First we have genus, then many genuses are divided on the basis of species. After species, like species is still this much, like Puccinia is the genus. There are so many Puccinia. Like after Puccinia we have graminis, means it is for grasses, right? Gramini family. Then we have the species. Graminis is the species. After Puccinia graminis, genus and species, we have this forma specialis, variety or special forms, which is this triticii, hordi or aveni. Then they are subdivided into forma specialis. After that, different varieties of symptoms. Based on different symptoms, they are subdivided into race. Again, based on the localized field population, they are divided into isolate. Further, on the basis of clonal population, they are divided into single spore derived biotype. Okay, so this is for fungi. For bacteria, it's the same thing, only in case of former specialists, we call it pathover. Okay, and for viruses, instead of this races, we call it strain. So yes, organism to pathogen it varies, but almost it is the same thing. At start it starts with the same, but sequentially it differ a little bit. Okay, so the whole paragraph you can understand with a single table, or if you learn it. Okay, types of plant resistance to pathogens. In nature and crop production system, we observe a great variety of disease susceptibility and resistance. This is largely explained by genetic compatibility or incompatibility of host and pathogens. Additionally, environmental factors influence host and pathogen such that environment genotype interactions contribute to the occurrence and the severity of diseases. Furthermore, some crop genotypes can tolerate a certain degree of disease and still produce a good yield. Yes. The genetic complexity of resistance. One such example I'll give you here about this, like some crop genotype can tolerate a certain degree of disease and still produce good yield. One such example which is coming to my mind is of citrus canker. It is very harmful, right? But apart from the scenario that it causes citrus canker, it is not mycotoxin. So even if we get to see it in the fruits, but we can still consume. We should not, but still if the disease is minor or small, then we can consume it, and it is marketed. At least in India, we will visit a lot of market, and you, by seeing, you will understand the symptom that it is citrus canker, but even after that people buy it and they consume it, and nothing happens because it is not mycotoxin, means it's not harmful to the humans. Okay, so hence breeding and biotechnology for disease resistance remain a major task for agronomists and should be informed by the virulence potential and reproductive lifestyle of the pathogen. In general, we observe resistance to non-adapted pathogens, incomplete race non-specific resistance, and pathogen specific resistance, which is often complete. Okay, resistance to non-adapted pathogens. What is it? To be successful in causing disease, pathogen must be adapted to a certain range of host species, as outlined above. Other plant species, when confronted with such pathogen, usually resist the infection attempts and show full resistance without any symptoms. Yes, same concept. They have just repeated it. Okay. Non-host pathogen, this is the thing. This can be the result of a failure of a pathogen to express a proper virulence program on the particular plant because it lacks certain receptors for sensing the plant as a potential host. If Puccinia target potato, then or Puccinia is a rust pathogen, and Euromyces is a rust pathogen. For example, Euromyces causes rust in beans and the pulses. Even if both the pathogen Puccinia and Euromyces, they are rust pathogen, but Euromyces, if it attacks wheat, it will not cause the disease. Similarly, if Puccinia attacks the pulse crops, it will not cause disease, right? So this is host specificity. Simple. However, in many cases, non-adapted pathogen actually sense the pathogen as a potential host and attempt to infect but fail to overcome preformed and pathogen activated defense responses. That's it, you know. Not to repeat the same concept again and again. Okay, last one was resistance to non-adapted pathogen. Now, res, non-specific resistance and qualitative trait locus. Let's see what they're saying. Pathogens can adapt to their host plant in a long coevolutionary process or by so-called host jumps when they suddenly acquire or lose certain function to overcome incompatibility and gain virulence. Yes. Now what happens, for example, there are, I told you, there are few sequence, okay, which makes them capable of targeting a particular host crop. Now in case there are some changes, okay, let's do it here. Okay, for example, for Puccinia we have A, T, G, C, A, T, G, C. This is for Puccinia. Okay, this is for Puccinia, and the sequence for Euromyces, for example, it will be A, C, G, T, A, C, G, T. This is for Euromyces. Okay, by any chance, if something happens, there is some deletion, substitution, addition, whatever it is, if the changes of this Puccinia, it got changed, and it becomes like Euromyces, the base sequence, it get changed due to mutation and recombination. So what will happen? It will overcome incompatibility and gain virulence. So Puccinia, which was before, it was not virulent to the pulse crop, it will gain the ability to attack the pulse crop. That is it. Okay. So first it was incompatible, but now it will be compatible because of this mutation and recombination. Okay. Such an adapted pathogen show basic compatibility with a certain plant species and cause symptoms on that species. However, not all host genotypes show the same degree of susceptibility to the corresponding disease. When we observe complete resistance or single host plant genotypes, that is usually restricted to certain races of an adapted pathogen, it's called race specific resistance. Okay. Okay, let's come to race specific resistance directly. Okay, so what is race specific resistance? It is a striking observation that in a pool of genotypes from the same host plant species, some show complete resistance to certain race of an adapted pathogen. Simultaneously, the pathogen that cannot infect that plant genotype can infect other genotypes of the same plant species, and the host genotype that shows complete resistance to one race of the pathogen can be very susceptible to another race of that pathogen species. So what they are saying is that if we have 3 races of a single pathogen, okay, now consider this genotype of a host. If this genotype is resistance over race A of the pathogen, which does not mean it is also resistance of this races of pathogen. Consider this is example of Puccinia graminis triticide res A, Puccinia graminis triticide res B, Puccinia graminis triticide res C. Okay, and this genotype of host is completely resistant to res A. Even after that, it will be obviously susceptible against these 2 races of the same pathogen. Understood? See, in this table we have seen, right, in this table, like a pathogen having same genus, species, former specialist, even after that they can have separate races. They can be differentiated among different races based on the symptoms. And here we have seen some examples like race 1, see like Puccinia graminis triticei, race 1, race 15, race 13. So the example was as such, if that host is totally resistant for this race 1, but it will be susceptible to race 15 of Puccinia graminis triticei and other so on races. Okay, I hope I could make that portion clear. From this point we will come across few of the terms such as disease escape, disease tolerance, right? So the advanced environmental conditions harm not only the host but also the pathogen. Hence, genetically susceptible plants can appear resistant because environmental conditions do not support pathogenesis. We have discussed about the disease cycle, right? Disease triangle, right? So in the triangle, what are the basic requirements that is required? The first is susceptible host. The second is virulent pathogen. What is the third one? Third one is favorable environment. This is it. So even if the disease is causing the pathogen is attacking the susceptible host, even after that the disease will not occur. Why? Because the environmental condition is not favorable for the pathogenesis. Okay, I hope the term is clear now. So I'd like to give one simple example and move on with it because the video is getting too long and there is nothing hard and tough rule or definition of disease escape. Just the one I told you. So one such example is pathogen require high humidity and the present environment is not as such, like we're having very hot weather, okay, like June, July, September that we are having right now. So the humidity is not that high, so the disease will not occur. Coming to disease tolerance. Yes, tolerance to disease. So what is disease tolerance? Means disease will cause. In this case, disease tolerance to disease is the ability of plant to produce a good crop even when they are infected with a pathogen, which means the crop is infected by a pathogen, but even after that the disease is tolerable and it will produce a good crop. That is disease tolerance, means it is tolerable. That's it. Okay, the pathogen survives but the plant shows little damage, not too much to cause severe loss. That's it. Moving on, we have plant pathogen co-evolution. Co-evolution means what? Means both are evolving together, right? So here the plant and pathogens are in constant battle, adapting each other. So what happens here, like when plants get new resistant genes and pathogens evolve new virulence genes and vice versa. So it can be explained with the help of two models, which is one is arm race. We will see here, yes, one is arm race models and the other is trench welfare model. So what is this arm race model? Arm race is the continuous cycle of new resistance and new virulence, but it is unstable. So what happens in arm race? It describes a process in which interacting effects of plant and pathogen genes frequencies lead to boom and burst cycle. This is one of the example of arm race model. So what happened in boom and burst cycle? The graph actually looks like this: boom and bust, like a mountain. Okay, so what happens here? Boom is the what we call in Hindi like Ashirvad and all. Okay, the burst is the curse that we get. So how do we explain it? Is when farmers adopt a new crop variety that possesses a powerful resistance gene against a specific plant pathogen. For example, rust. I have been taking example of rust in this whole chapter, so I'll take rust again. But what happens? The variety becomes highly popular and widely planted because of what? Its effective resistance capability. Okay, so this is the boom. Okay, the crop is highly resistant to that particular pathogen. But what happens next? A new strain or pathotype of the pathogen evolves that can overcome this specific R gene, which is responsible for the resistance. So this new variant strain spread rapidly through the pathogen population, causing the resistance in the widely crop variety to fail. This is the burst, okay, where the crop becomes highly susceptible to that disease again, resulting in losses. So it happens boom and bust, okay, gaining resistance and cultivating it worldwide, and because of which the pathogen recognizes the resistant gene, and it what a new strain, virulent strain spread, or it develops and it spread rapidly, making it leads to burst. Okay, so this cycle repeats again and again. That is boom and bust cycle. Okay, moving forward, this the breeding for plant disease resistance. So what they are saying here is that the value of plant resistance in controlling plant diseases was recognized as early as the beginning of the 20th century. The obvious advantage of planting a resistant instead of susceptible variety made the effective breeding of resistant varieties possible and desirable. Okay. Okay. So what in the breeding, there are many new technologies came, which modern tools that we get to see nowadays in the present time, which makes breeding like faster and more precise. Some example of those are like mass. We have heard of mass. I'll write it. I can't find it here. One of such example is mass. Mass means what? Marker assisted selection. Okay, this is DNA based totally. And then we have like genomic selection, and then CRISPR. Yes, genome editing. CRISPR is very common nowadays. CRISPR-Cas9, all those. Okay, so the breeders combine like the high yield and the quality and resistant in the new varieties, which makes it more stable. They give us stable yields even under the disease pressure. That's what the plant breeders are doing nowadays. Without delay, we will see the breeding for vertical or horizontal resistance. This is very important. Sometimes many questions come in like broad questions. We see difference between vertical and horizontal resistance, or separate questions can come. So I'll just say it. You can note it down. Okay, I don't have a pen and paper right now, so okay, I'll just saying it. Like vertical resistance is monogenic and it's race specific, which we have seen in the earlier section. Yes, and it is controlled by one or few major genes. Which type of genes? R genes, the resistant genes. Okay, and it works only against specific race of a pathogen. That is why it is called race specific. Okay, and it follows gene for gene concept given by whom? Floor, right? Okay, don't forget it. And it can make plant completely resistant. It is called complete resistance, whereas horizontal was partial. I'll come to that point. Okay, there are few limitations of vertical resistance, like it's short-lived and it often breaks down when pathogen mutates or new race appears, new race of pathogen appears, and it has a high risk of sudden epidemics if resistant fades. Okay, coming to horizontal resistance, see, there are effective points are given for a vertical resistance, like it is incorporated for the annual crops, and it is directed against pathogens that do not reproduce and spread rapidly. Okay, remember my points that are easy to understand. Apart from this, in contrast, we will see about horizontal resistance. So it is incomplete or partial resistance, whereas vertical was complete, and it is durable, yes, and the vertical, that one was not durable because it is short-lived, whereas this horizontal is durable. Yes. And what more am I missing? Okay, the vertical was race specific, and this horizontal is not race specific. It has a wide or broad host range, and it is governed by minor genes. It is actually polygenic and race non-specific. Okay. Coming to disadvantage, we have discussed about few of the disadvantages of vertical. So for horizontal disadvantages, it has protection. The protection of horizontal resistance is weaker compared to vertical resistance, because the target is, so many targets are there for horizontal resistance. So yes, the protection is weaker, but it's more durable. And for vertical, the protection is strong because it is very specific, but the durable is very less because the resistance can be overcome by a new strain of pathogen. We have one last topic to cover before we wrap up this chapter. That is sensor-based technique to assist plant breeding. Sensor-based technique, this is the new technique that we are using nowadays. We are using, or it should be used, for fast detection. Okay, with the onset of sophisticated remote sensor systems applied to monitor plant growth in the field, also scoring for plant disease became feasible to support the identification and selection of resistant plants. Now, remote sensing can be based on visible light, okay, sensors, near infrared light, shortwave infrared, and thermal spectra. The sensors used are composed of combinations of multiple wavelengths appearing in this visible RGB, which is the visible light, video cameras, multiple cameras made up of several selected NIR wavelengths. NIR is this near infrared, or hyperspectral cameras, multispectral, or hyperspectral cameras. Hyperspectral is with several hundreds of wavelength bands across the visible light, infrared light, or shortwave infrared range. Also, automated fluorescence sensors used under the microscope combined with sophisticated software algorithms to calculate the percentage of infected cells or leaf area can be found. The sensors are stationary, placed on field rover or mounted or unmanned aerial vehicles. Unmanned aerial vehicles. With this, you can think this is something different, but you know, this unmanned aerial vehicles, this is also, we can call it drones. Yes, drones we have heard. Yes, that is also called as unmanned aerial vehicles, UAV. The selected sensors and vehicles depend on the required spatial resolution and temporal throughput. To identify and select resistant plants, say in visible light, this is how sensors works for multispectral, hyperspectral, thermal, fluorescence, and 3D sensors. Okay, to identify and select resistant plants, let's see. Free of pathogens, remote sensing devices are applied to resistance scoring, indicating the degree of pathogen appearance on the plant organs like stems, leaves, inflorescence, or fruits. The scoring may take place, for example, inside an infection hood after a controlled pathogen attack, or in the field under natural pathogen infestation. Repeated measurements over a time series allow to quantify and model the temporal progression of the disease. To date, RGB cameras and multispectral. Let us quickly check what is this RGB. Okay, RGB camera full form. Okay, so this RGB is the colors like red, green, blue, which refers to the primary colors of light used by the camera to capture color images. Okay, now we know what is this RGB. And multispectral camera may be the most popular remote sensing devices used in applied breeding programs to select resistant plants. Of course, the image data have to be processed and modeled using machine learning routines. This can be carried out either in-house by hiring IT specialists or commissioned to external service providers. I will suggest you make friends with the people studying machine learning and artificial intelligence. They can help you work with this early detection of various diseases. Nowadays we are using it for the early detection of viral diseases because it's difficult to detect and diagnose the plant viral diseases. So okay, that's what it. And it was quite different from normal plant pathology that we study, because here we have heard so many terms from genetics and plant breeding, but it was interesting and it was totally relevant. So okay then, I hope you understood it. If any doubt, please comment down, and if any suggestion or recommendation that you have, please feel free to tell me. I'll try to improve myself. Or you have any recommendation on my pedagogy or the way I discuss, so please let me know, and I'll try to improve it in my from my next video. And any suggestion is highly welcomed. So we will be back soon with the next chapter and with something new and interesting. Till then, keep learning, keep growing. See ya.